Welcome to Caitlin and Hannah Biology. Today we'll be talking about translation and transcription. These two processes are how we go from DNA to RNA to proteins. This transformation is called the central dogma of biology. We start out with DNA that is found inside the nucleus. It contains sections of genes that we need to code for proteins. We then copy down the gene to make messenger RNA. This process is called transcription.
The messenger RNA moves out of the nucleus and into the cytoplasm, where it attaches onto a ribosome to make proteins. This process is called translation.
To remember the order of these two processes, you can think of the alphabet. Transcription has a C in it, and translation has an L in it. C goes before L in the alphabet, so transcription goes before translation in protein synthesis.
Transcription. In a eukaryotic cell, we will find double-stranded DNA in the nucleus that, when you uncoil it, contains sequences of nucleotides, which includes adenine, thymine, guanine, and cytosine. RNA polymerase is an enzyme that will bind to the DNA at the promoter region and move along the strand, transcribing the DNA into RNA. In DNA, adenine and thymine are paired, but in RNA, adenine pairs with uracil. So every adenine on the DNA strand being transcribed by the RNA polymerase will have the complementary uracil base instead of thymine. This new copy, produced by RNA polymerase, is called messenger RNA or mRNA. The mRNA will be altered by the addition of the 5' cap in poly A tail, which is the addition of adenine bases to the end. There will also be sections of the strand that do not code for a gene, which are called introns and are removed. The final mRNA strand that has the message to make the protein ends the process of transcription.
Translation. mRNA will move out of the nucleus and into the cytoplasm, where it will attach to a ribosome where the protein will be made. This ribosome is made up of proteins in a form of RNA called ribosomal RNA, or rRNA. The ribosome is made of a small subunit on the bottom and a larger subunit on the top. As the mRNA flows through the ribosome, every 3 nucleotide bases on the strand will code for 1 amino acid. These 3 bases are called a codon. The amino acids are found on top of one another from an RNA called transfer RNA, or tRNA. tRNA transfers its amino acids from the cytoplasm to the ribosome. tRNA has 3 nucleotide bases on the bottom called the anticodon that will bind to the codon found in the mRNA. Then it will release its amino acid, which will be added to the expanding polypeptide chain that becomes the protein. tRNA then flows out of the ribosome and into the cytoplasm to get another amino acid. This process continues until there's a stop codon, which ends the sequence and completes the protein. The proteins that are created can be structural, which could be forming a part of the cell membrane that helps with transport, or the protein can be functional, which means forming hormones, chemicals involved in cell chemistry, and enzymes that speed up chemical reactions. When figuring out what 3 nucleotide bases code for each amino acid, it is smart to look at a codon chart. This chart represents the standard genetic code for an RNA codon. There are 20 amino acids, but 64 possible 3 nucleotide base codons. Therefore, some amino acids can be specified by more than one codon. Understanding how to read a codon chart can be a little confusing at first, but with these easy steps, you'll be able to figure out the amino acid that matches with your codon in no time. Let's use the codon AUG for an example. The first step is to locate the first letter of your codon using the left side of the table. Ours is A. The second step is to look at the second letter in our codon, which is U. Find the second letter at the top of the table and follow it down the chart until you reach the first letter from the left side. So our second letter is U, so we'll go down the left side until we reach A, which is our first letter. You should see 4 amino acids. Step 3 is to look at the third letter in our codon, which is G. On the right side of the chart, find the third letter G that will intersect with the box that had the 4 amino acids. Move your finger from the G over to the left, where you should land on methionine. Methionine is the amino acid that the codon AUG matches to. You can use these 3 steps to find any amino acid for any codon. The bases AUG represent the amino acid methionine, which is the start codon that begins the polypeptide sequence. The bases UGA, UAA, and UAG do not code for any amino acid and serve as the stop codon and represent the end of the polypeptide chain.
These proteins that are made are responsible for the color of a flower, the shape of a leaf, the blood type of a human, and even the sex of a baby. Proteins are a type of enzyme that speed up and regulate chemical reactions. For the color of a flower, there's a gene that codes for the enzyme that produces the certain pigment. Genes that are for certain proteins are able to regulate organisms' size and shape. Proteins construct and control factors in the cell.
Thanks for watching. Hope you learn more about transcription and translation.